Okay, Jan Ankersmit from Vienna, surgeon, not that differentiated, not a basic scientist, but applied scientist. My talk is Secretome-based regeneration from heart to skin, and my issue for this Congress was to show a pathway how to get a medication into humans. And as we had this logo, we said from bench side to the patient, and that involves the regulatory affairs. And as you will see in the next session, these are the people, the regulatory body who brings the treatment which we describe in the research into clinics. So I advise you to see this. So let's start. So how stem cell therapy works. This was as you have heard now, these two talks by Professor Wollert and Neki with the stem cells, hematopoietic stem cells. This is not the story. People tried to separate stem cells, and these stem cells were injected into coronary or into the heart. And basically the idea was that these stem cells are going to transdifferentiate, and now we believe that this is not the case. But we see an effect, not too much, but we see an effect. Then in 2015, a colleague of mine, Professor Göngeschi, a cardiologist here in Vienna, she published the CURE study. And what did Marianne do? She went to all centers who performed stem cell therapies, asked for the data sets, and then did, with a statistician, she looked at the raw data. So it was not a meta-analysis, but she really looked at the raw data. And she published in this journal, in the Circulation Research, that there is no effect of systemic intercoronary infusion of stem cells. So basically that what was said here on this podium was confirmed in this publication, and that even got one of these very prestigious HA circulation rewards. So that is the knowledge of 2015, and that's a take-home message. Stem cells in the heart, into coronary, this is futile. Then that's why my friendship with Professor Gnecki, he was the first to show that mesenchymal stem cells and the secretome, the secretome which was produced by mesenchymal stem cells, has an effect and prevents, in small animal models, prevents myocardial infarction. And this was also advocated by Professor Wollert, that basically the secretome of stem cells is these paracrine factors, as it's called, so you can call it secretome or paracrine factors. They are cytoprotective, and they have lots of functions which are believed to help the heart to not have the infarction as it would deserve. So this was suggested by Professor Gnecki from Pavia. Was done in his postdoc time at Duke University. 2005. We are still aware that there are some stem cell trials ongoing, but as it is already published extensively, this game is over. Here I want to advise to read one nice paper by the senior author Menashe. Menashe was one of the first authors who basically took muscle cells and injected them into the heart, and has shown that, and that was published in The Lancet, I think, at that time, that was sort of the initiation of the hype that these transplantation of these cellular compartments increased heart function, as far as I can remember. This was then followed in Nature Medicine paper in Nature, where they took bone marrow cells, and they also showed that there is in some rat models that these hearts are doing better also in mice. And if you look at this is basically the sum up of somebody who was working for 30 years in the field. We know this metaphoric picture, and he relates it to myoblast, then going to bone marrow cells, ending up with some cardiac spheres. And he, in his review article published just in 2015, said, well, the secretome is the answer. So in the world of clinical reality, this is fact. I thought I really advise to look at this paper. So coming now to my research, first notion. We are talking preclinical research. Then we have got the regulatory affairs, which basically regulates what is going into the patients. Then you've got the clinical research, the proof of concept, and then you've got the application with the humans. So if you talk and if you listen to many of these congresses, you most of the time see a lot of research which is just pertaining to preclinical research. Preclinical research is that what most of the young people are doing here in this room. They are having an idea, some PIs telling them, well, this is a good idea, please go on, follow this path. This is mice murine models. And then if it comes to the translation, it becomes very expensive. And for an applied researcher like I am, as a surgeon, this is sort of like, that here becomes very, very expensive. And that's beyond any realm of a research facility. However, these are the people who have the ideas. So if a society says, if you want to augment the value of research for society, there has to be in the funding agencies something that's going to cover this development, because now at the moment this is futile. There's nothing in existence. So if you write grant applications, it's always about preclinical research. This here is left over to big companies. And as I am now understanding, big companies, they are most of the time not at all interested in investing into new therapies. That's just my personal notion on this. But so first, I'm going to show you preclinical research, which ends up in a lot of papers. So that's lots of. It's a bit condescending what I'm saying. This is paperwork. So paperwork. This is just a few of the papers we have published in the last few years, where we have investigated the secretome of white blood cells, and you're going to see and hear the talk of my colleague Dr. Michael Milner. In the last year, we're going to talk about the mechanisms of actions. But basically, this is here. Lots of paper, but it's just something, it's an ego booster. Ego booster that doesn't lead to anywhere. So this is a harsh statement, and that's why we have to think as basic scientists, translational scientists, how to augment and make awareness with politicians to augment clinical research. Okay, let's continue. So what I'm talking about. Some of my friends here have seen this slide extensively. I just have to repeat this now here. If people have shown that stem cells, if you infuse them in rodents or in mice, into myocardially or into the systemic venous flow, then you can prevent myocardial infarction. So when in 2003-04, I had this idea, I looked at this and then I got a bit angry, because I said, well, there's a control experiment missing. And then I just did the following. I sacrificed mice, took white blood cells, so not stem cells, but our white blood cells, which are easily obtainable. I irradiated them and then took this cocktail, so the paracrine factors with the white blood cells, and infused them intravenously into a rat that underwent myocardial infarction. And then we have seen here this effect. Basically, the suspension of apoptotic white blood cells, not stem cells, preventing myocardial infarction. And that was published in 2009 in some minor paper. Anyway, this wouldn't have been accepted in any other journal. So, take-home message. Take-home message. What was the take-home message? Let's discuss this. So we have got here paracrine factors, or the cellular compartment. So then we had this hypothesis, which was based on that what we saw on the bench, that it must be the paracrine factors of white blood cells that do the regeneration. So now I'm coming to the application aspect. Application aspect. If you separate the cellular compartment, you are left over with the paracrine factors from white blood cells. And what you can do, and this is something what you know from Nescafé, it's lyophilized powder. You can lyophilize this and you can say this is our drug compound. Based on this idea, I received in 2009 a Christian Doppler laboratory. My my thank you to this society, which make the following work possible. So just go to show you how it should work or how it works now at the moment. So if you donate blood, and there's for sure some people here in the room who donate blood, you donate your blood, and then you take the red cell compartment for transfusion, you take the plasma for antibodies or for factors, and the white blood cells are discarded. So what we did, we separated this, took the white blood cells, irradiated these white blood cells for 24 hours, and then you centrifuge, so there's no cellular transplantation anymore, you have just got the paracrine factors. And then there was an issue which I didn't know, because it was completely new land for me, and we had to, because of regulatory affairs, we had to do a double viral clearance. And that was then our drug compound that can go into humans. And that was around 2004 to 2005 developed. So coming to our talk, Marian Göngeschi has seen our rodent model. And Marian is an experimental cardiologist with a huge cardiac facility in Hungary. And then we did the following. We infused the paracrine factors after AMI after 40 minutes. And we were able to show that we could prevent myocardial infarction in large animals. So I'm saying if you want to translate something into humans, you have to do the rodents, then you do the pig, and then you go into humans. So then my colleague, Dr. Hötzenegger, thoracic surgeon now at the moment, he has shown in the European Heart Journal that in 2013 that if you induce in mice myocarditis, you can basically, with one injection intraperitoneally, you can completely abrogate the inflammation. That was done with Professor Eriksson, and Professor Eriksson will give a talk, I think, tomorrow. And then that's the next speaker, that's Michael Milner, that he showed in a wound healing model with murine. In a murine model in 2013, he showed that basically if you put the paracrine factors into a cream, you can augment healing. That was published in 2011-12, so some of that is known, and this is preclinical work. And at the end, this was the suggestion by Professor Schachler, well, if it works in murine model, please perform the same experiments in large animals. And so my colleague, Dr. Hacker, he did this experiment where we burnt the skin, did the excision, and here I have to thank Professor Redl, who's in the room, who basically taught us this experiment. And then we did the split skin, and then we treated up to 10 days, as far as I can remember. Yes, I can remember well, yeah. And we were able to show that the paracrine factors from apoptotic white blood cells increased wound healing. It's a huge issue for plastic surgeons and burn wounds. So take-home message, in a preclinical setting, we were able to show that paracrine factors from white blood cells are doing systemically and topically something good. That's the story with the preclinics. So next step. Here, of course, as a surgeon, you have a tumor, as in my case, if it's a tumor in the upper lobe, you resect the upper lobe, do the lymph nodes, and then the patient is healthy. This is my approach towards medicine. So then it took me some convincing arguments, and there's one person in this photo not available. This is Primarius Gabriel, the former head of the Rote Kreuz in Linz, and this is the current leaders in blood bank in Linz. And this is Ms. Süssner, Mr. Watzinger, and Mr. Jüngling. They have supported the idea to produce this paracrine factors under GMP conditions. So basically, if you have a product, you have to produce it under GMP conditions, and we're going to hear much more by Anja Peterbauer. So take-home message, drug compound, we see it in preclinical, that's working. Then the next step is the GMP facility, and then you go into the clinical trials. What is then the next procedure? If you have proven the GMP facility, you get an audit by the regulatory affairs, the Austrian Health Authority, and they have approved that basically everything what we are doing is correct. And this basically helps the society that they don't get any treatments that are not correctly performed. As a next step, we then did a we wanted to know whether these paracrine factors are doing harm in healthy humans. And so we, with Professor Wolst from the KKS and from the clinical pharmacology department, we did the first secretome-based study, which was a double-blind controlled. After the AGES has approved our clinical trial. So here we've got the validation of the process, we've got the okay from the AGES, the regulatory affairs. Then we had this clinical pharmacology experiment in humans, and we were able to show that our secretomes are not doing any harm to our patients. So now, as somebody in the research, you start to imagine how much work is involved for this. That was done between 2014 and 17. So this was published in scientific reports. So as I perceive it, it's maybe the first secretome-based therapy, which is published double-blind, which was a ClinGov number. Let's take it as this. But the only thing what really is important in this setting is that and that was done always with the stem cell therapies. The patient has an acute myocardial infarction, comes in the hospital, the autologous stem cells are retrieved and then are injected into the patient. So this was here the same case. So we took paracrine factors from the patient. So we had the proband running or driving to Linz, got by the team of Ms. Peterbauer the autologous APOSEC produced, and the APOSEC was then delivered to the pharmacy, and from the pharmacy it went to the clinical pharmacology, and there this study was monitored. So basically a huge work. Autologous therapy is, however, unfeasible. It's not feasible. As we have heard, long duration, you are very late in the disease process, so this is no option. And also for a company who wants to develop something, this is here something what you can reject. It's nice for research, but so we had to go another way. So here we see how it works in Linz. If I, as Jan Ankersmit or somebody of you, goes to Linz, you get your white blood cells retrieved. And then after about 48 hours, you get your autologous APOSEC, which cannot be too much because you can just donate half a liter of your blood to 400 milliliters. So basically, you don't get really a sufficient number of paracrine factors. So we had then this idea of pooling our paracrine factors from multiple donors. So you produce from multiple donors the paracrine factors, and then you just put them together, but then you have a real problem with the regulatory affairs because they say this is a cell-derived biological. So this is now for the regulatory affairs, it becomes a completely different story, which is nearly impossible to overcome. I hope I can convince you that we did, but basically, if you take it's now German, sorry, you take the soups, as Dr. Nicki said, you take the soups, pour them together, then you've got the allogeneic APOSEC, and this has to be then termed as a cell-derived biological. So making now the difference, this is now something which has to be said. We're talking in some talks here about ATMPs. They are living cells. They have more or less many mechanisms of action, and they are approved by the regulatory affairs. But, and this is the most important thing, you cannot do any viral clearance with this drug compound. So these are the cells which are sort of like coming from super batches, but here we see there's no security for the patients involved. If you go now to our drug compound, it's produced by living cells, by white blood cells, which is basically now at the moment the current waste product. It is multiple paracrine factors, so it means, as with the ATMPs, it's quite eclectic, lots of mechanisms of actions, and you can do a viral clearance. And then it becomes very expensive because then you have to secure for the regulatory affairs all issues which include toxicology, viral clearance. You have to prove viral clearance is working, and at the end you can start them with a phase 1 or phase 2 study in one indication. So this is something which is really very far beyond any preclinical work. Here, I'm nearly finished with my talk. Here it becomes now mandatory to talk with the regulatory affairs. Me as a researcher, applied researcher, cannot do this job anymore. So we found a company, which is the Dr. Regenerative GmbH from Badenweiler by Freiburg, and they are helping us to discuss our topics with regulatory affairs. We have received quite comforting resonance from the AGES, and next week we're going to go to the PAI, the Paul-Ehrlich Institute, and then we can continue talking. But basically, APOSEC is a non-cellular treatment-based paracrine factors. These paracrine factors have to undergo toxicology experiments, so we have to prove to society that our drug compound doesn't do any harm. You have 5 species. We have now 5 species with our preclinical program, and in Hamburg, a very prominent German provider, CRO, is doing our toxicological program. Here I also have to allude to consultants who are helping us with this project. Without toxicology experiments, no drug gets into the human. This is fact. We all know, the older people here in this room know the Contergan scandal, which was basically the initiation that all drug compounds which are used have to go into toxicological experiments. So, just to sum up this whole story, at the end, how does it look like? We have the regulatory affairs, which say, well, this drug compound is produced well. Please listen to the next session. Then you go with the project to the ethics committees at the medical university with the complete program, which is ascertained by the clinical pharmacology. The ethics committee at the university gives its okay. Then this whole approved project goes then to the AGES or to the PAI, and they have to also approve this clinical trial, and then we can start to do our clinical trial. So that means now, and this is now basically the sum up of this talk, preclinical work is fine. If you want to get ahead for the patient, you have to finish off preclinical work, which is toxicology, potency assay, etc., and then you can go into the phase 2 proof of concept trial. So what I'm saying now is the following. Compassionate use of a new drug compound is no value for society. This is just some nice photos, and these are case reports. You can publish this possibly, but it has no relevance for our society. Second thing is you have to perform a proof of concept study in one indication, and then if you have shown this, you can start with the phase 3 trial, and then you get the approval by the EMA. The next slide is just a thank you to all these people who supported these efforts, especially now my host of where I'm putting my work in is the medical university, the company ApoScience, and the FFG and the Christian Doppler laboratory. And of course, my department had the thoracic surgery unit where I'm working about 40%. Thank you very much.